Dr. Lee holds a master of bachelor and a master in mechanics and engineering science from Beijing University, as well as a PhD and a postdoc training from UC San Diego in bioengineering area. So Dr. Lee's research is focused on cell and tissue engineering, and that he has contributed to the understanding of how biophysical factors regulate stem cell differentiation and cell reprogramming. His multidisciplinary approach has led to significant advancements in the engineering of biomaterials, stem cells, immune cells, and pushing the boundary of the tissue regeneration and disease therapy. So Dr. Lee's extensive research has been featured in some of the most respected scientific journals, including Nature Materials and the Nature Biomedical Engineering. And his innovative spirit has also translated into 16 patent applications. So for his extraordinary contribution to bioengineering, Dr. Lee has been recognized by his peers and the scientific community at large, earning the prestigious honor such as the Fellow of American Institute of Medical and Biological Engineering and the Biomedical Engineering Society. So today, Dr. Lee will share insights on the intriguing interface of mechanics and genetics in a seminar titled Mechano Epigenetic Engineering for Cell Reprogramming. Please join me to welcome Dr. Song Lee.
I want to first thank Dr. Lee for a kind introduction, and thank all organizers for organizing such a great event. And particularly, I want to thank Professor George Diaz for the invitation, and it's great to meet you and the team at UCLA a couple of months ago. And we already have some connections, several projects, for example, the AHA projects, the Engineering Research Center, supported by NSF. We look forward to more collaborations in the near future. So today I want to actually expand the perspective a little bit and talk about two stories on mechanobiology and for cell engineering. For those who haven't been to UCLA, here shows a part of a beautiful campus. The engineering building actually is not here, but towards this corner. At UCLA, we had a long history of engineering development, and these started in 1960s and 70s, while mechanical engineers worked with orthopedic surgeons to develop orthopedic implants. And also, you probably know that PET imaging was invented at the UCLA by engineers who worked with medical doctors. So around year 2000, bioengineering department was funded, as many other universities in the past 20-25 years. And we carry on this interdisciplinary research program built on the graduate group we had before. And we started undergraduate education, and we really benefit from the top engineering and medical center on the same campus. That's very, very helpful. And you can see from the composition of our faculty, we have a core, joint, and affiliate faculty, almost 100 faculty in our graduate program. And they are from various disciplines in different schools: engineering, medicine, dentistry, life sciences, in physical sciences, etc. And we also have adjunct faculty in industry. In bioengineering, we divide the research areas in these 5 sub-areas. One is molecular, cellular, and tissue engineering. This is the largest research area, and that includes many different small areas, such as drug delivery, stem cell engineering, biomaterials, molecular probes, etc. And that interfaces with traditional biology and chemistry. The second area is biomedical devices and instrumentation. That ranges from wearable devices, micro nano devices, up to very big instrumentation we use in the hospitals. The third area is biomedical imaging that integrates hardware, software engineering together with graphics, etc. To help clinical diagnosis. In a very fast-growing area is biomedical data sciences. That is also very broad, including systems biology, bioinformatics, precision medicine, etc. And neuroengineering is one example that we integrate all of these technology platforms and use that to study the brain-machine interface and also promote the regeneration of central nervous system and peripheral nervous tissues. My research area is in the first area here in cellular and tissue engineering. So on one hand, we're interested in understanding in vitro and cell culture system how biophysical factors together with chemical factors and biological molecules and regulate cell functions. And this is half of our lab in vitro work. And on the other hand, we're interested in translating what we learn in vitro and integrate this with various other approaches, for example, drug delivery, materials engineering, stem cell engineering, etc., and use that for tissue regenerations in disease therapies. And currently, we are working on a couple of tissue systems. One is cardiovascular system, the other one is neuromuscular regeneration. And for disease therapy, and we are working on cancer therapy and diabetes therapy. And I'm going to show you a couple of examples today. And we know that in our body, at every level, we have mechanical forces. Right? So here shows the system, organ, tissue, cellular level, and down to molecular level, showing the active myosin contraction. And this example is a heart, cardiac muscle. And our focus is at a cellular level, because cells are the building block of our body. It can collect all information in the tissue microenvironment, sense this, and react to all of the microenvironmental cues, and remodel the tissue and organ, repair the damaged tissue, and can be used to cure diseases. So in the past 30 years, the area of mechanobiology has developed very quickly. And we have many examples showing here and summarizing this schematic. Around one cell, for example, in the cardiovascular system, you have hemodynamic forces that can result in shear stress on the cells, pressure changes, or even mechanical stretch in the substrate. In addition, we know that there are many passive mechanical cues around the cells. For example, in extracellular matrix, you have mechanical property difference, such as the stiffness or viscoelasticity. You may have experienced, for example, the micro-nano pattern in the matrix structure, or even the distribution of chemical ligands. So all of these factors could affect cell function in cells while integrate this information inside. When they migrate, proliferate, differentiate, or form certain tissue structure. So many studies on the short-term response of the cells. In a paradigm that people have figured out is that cells will respond to environmental cues through the surface receptors. And then the signaling molecules carrying this signal all the way down into nucleus through transcriptional factors and regulatory expression. And there could be very quick responses, could be seconds to minutes. And in the past 10 years or so, we are more interested in investigating the long-term responses of the cells that result in the cell fate changes, for example, stem cell differentiation, or cell reprogramming, or immune cell functional changes, etc. So I want to show you two stories. One is on the cell-cell interaction, how mechanical cues could affect the cell fate. And two is to engineer the genetic, the epigenetic state of cells and change the cell phenotype. Okay, so one example I'm going to use is the immune cells. Immune system actually not only are sensitive to chemical and biological molecules, antigens, but also could respond to mechanical cues. And here just show a few examples. For example, immune cells, when they are rolling, migrating, or going through the cell junction, getting out of the blood vessel through the actualization stage, you can see the mechanical interactions between cells. And for macrophages, in here in the second row, choose some examples. For example, they can deform in the 3D matrix during the polarization process, and there will be mechanical property changes, and even phagocytosis is a chemical process involved with cell-cell interaction. And today I want to focus on T cells in this study. And one thing that people know that is for T cell activation, the antigen-presenting cell could present various chemical signals to T cells, and T cell will recognize and develop specificity against certain antigen. In the past, we know that all these chemical signals are very important, and this is very important for various immunotherapy. And an example is the CAR therapy. So for cancer therapy, in the past few decades, it has evolved quite a lot. Right? We probably know the chemotherapy, the radiotherapy, etc., and those are not specific. In the past 10 years or so, and the clinical therapy has focused on more specific target therapy by engineering the immune system. And the chimeric antigen receptor T cell therapy is very promising and has been used clinically. It's still being developed. So this process involves the ex vivo expansion of patients' own T cells outside body for a couple of weeks. So first, you take the cells from the patient's blood, and then you expand them for about a week. And here, there's one thing we want to pay attention to, which is the activation signal presented by these dynabeads, which is polystyrene magnetic beads, very stiff, 1,000 times stiffer than the native antigen-presenting cell. So that's one mechanical difference. We want to get back to it. And then after a week, you introduce the CAR, which is the targeting signal to cancer cell. Through this viral transduction, and the cells will express this targeting signal. You expand them for another week, and afterwards you infuse them into the patient. So the whole process takes about 2 weeks. And this is a $500,000 therapy, personalized medicine treatment. Yet at this stage, this manufacturing process is not well controlled, yet to be optimized. So for example, when you introduce this viral construct into the cell, at the end, probably only half of the cells express this targeting signal. And many other cells do not have this CAR construct. And when you infuse all the cells into the patient, the therapeutic effect is compromised. In addition, for T cells, one thing we hope to have is they have the T cell have the long-term memory about these cancer antigens, so that you can have a long-lasting immunity against cancer cells. You don't need to infuse cells many times, right? Yet, when we have all these cells expanded before we infuse them into the patient, this population is a mixture, and the percentage of stem cells is quite high. Can we improve that? There are many things we can do. And here I want to show you the importance of mechanical property of this stimulatory signal. If you just simply change the mechanical property of these microbeads or microsphere, this could improve one, the viral transduction into the T cells, and two, we can increase the percentage of T memory stem cells for the therapy. That will increase the long-term immunity against cancer. So that's a short story I want to tell you. And this actually has been published as a preprint, is in revision or peer review publication. So this is the engineering part. We first make this artificial cell by using a very simple material, alginate, isolated from the seaweeds. So using this microfluidic system with the calcium cross-linking, we can generate a large number, tens of millions of these microbeads with very consistent size and composition. And we can do post-processing by cross-linking the alginate in two different ways. One is calcium-based cross-linking, which is reversible, and that will give the microsphere viscoelastic property, or you can use covalent bond to permanently link the alginate. In this case, provide elastic property. So we can manipulate the cross-linking concentration, molecular weight of alginate, and also the weights of cross-linking to control the mechanical property of these microbeads. For example, with certain viscoelasticity, and the relaxation could be 10 seconds or 20 minutes. In that case, 20 minutes is quite long. It's almost equivalent to elastic material. Or you can just make them purely elastic. So it is amazing what we found is that the cells are very sensitive to this viscoelastic property, and they respond to these, and during the CAR T cell expansion, we get totally different subpopulations. So she here gave you some idea how the cells interact with these different the beads with different mechanical properties. This is dynabeads, polystyrene beads. Here shows one T cell. They have interaction, but the synapse formation is not so much in comparison to the interaction of T cells or SynVac. We call it synthetic viscoelastic activating cell. You can see the synapse are trying to wrap around the SynVac from the T cells. And if you quantify the synapse formation by staining the CD3 epsilon on the surface, you can see there's more formation of synapse between the viscoelastic artificial cell and the T cell. So that shows the difference, mechanical property of the presentation of this antigen can make a big difference. And this is just a big picture. If you do single cell sequencing, after 2 weeks expansion of the cell, we see many subpopulations of these cells. And we don't need to go into detail, but I want to say that if you use dynabeads to expand the cells versus use the SynVac, you have a different percentage of all of these expanded T cells. For example, subpopulation 1, which is the CD8 positive memory T cell, increase if you have SynVac. And if you look into cluster 9, it's decreased. This is a fully differentiated effector cells. So our goal is to get more of the memory T cells so that we can have a long-term immunity, and the cells will carry on this memory for much longer. And if you use flow cytometry to analyze these cells, and you can see that we have a high expansion rate if you use the SynVac compared to control group, which has no signal, and also compared to dynabeads with the same chemical signal. In addition, it suppressed the regulatory T cell, which actually is not helpful for cancer therapy, to suppress cancer therapy. And another important thing is for CAR T percentage. We have over 90% of the cell expressing the CAR signal that can target cancer if you use SynVac expansion. So the mechanism is not very clear at this stage. It's a very interesting finding. That will also help the cancer therapy efficiency. And this is an animal model in a humanized mouse model with the B lymphoma model, the blastic cancer. And here we show 3 groups. Negative control without any treatment of the CAR T cells. These are CAR T cells expanded by dynabeads, and the CAR T cells expanded by SynVac. And here the color shows the tumor mass. The red color means a big tumor mass and going down to blue scale, and white means it's clean. And you can see here, without treatment, the cancer developed and the mice died after 28 days. And with the dynamically expanding cells, you have some clearance of the tumor, but these tumors come back because you do not have a sustained memory of the tumor cell antigen in these T cells. And yet with the SIMVAC expanding T cells, we have a sustained clearance of the tumor. So that shows that mechanical factor is very important for immune cell engineering. You can see the big difference. And this summarizes what we discovered for this story. If you are interested in knowing the whole story, you can look into our preprint published online. Okay, so now I want to talk more about mechanical epigenetic engineering, how we can control the epigenetic state of cells and manipulate cell phenotype. For various cell engineering approaches, in this circle summarizes most of these possibilities. For example, if you have pluripotent stem cells, such as embryonic stem cells or iPS cells, they can differentiate into multipotent cells and finally become terminally differentiated cells, such as neuron, cardiomyocytes, muscle, etc. And now we know that you could also take a fully differentiated cell, such as fibroblasts or any other cell types, and convert them all the way back to pluripotent cells, or you can directly convert them into other cell types. So here, today, I want to show you the example by using this model, by converting fibroblasts into neurons. And this process will take a couple of weeks, just like stem cell differentiation. But the beauty of this system is that the most important time period to determine the cell fate is in the first 2-3 days. So when you manipulate various factors, you can see whether they are effective and whether they affect the cell reprogramming process very quickly instead of waiting for 2-3 weeks. So one important thing I want to introduce is the epigenetic state of the cell. And we know in our body we have over 200 cell types, and all the cells have the same genome sequence. If you consider the genome sequence is the hardware of a computer, the operating system is the epigenetic state that controls the gene expression. Which genes turn on, which genes turn off. Although we know very well the genomic sequence, we have done the sequencing, that's all known, we do not fully understand how this operating system works. So that's one big puzzle in biology. But we do know there are a lot of commands in this operating system. For example, this biochemistry modification of the histone, which is a scaffold for the chromatin organization and also DNA itself. And they can change the organization of the chromatin. So in the genome, the epigenetic state will control the organization of chromatin, and we divide them mainly into two major types of structure. One is called the heterochromatin, which is closed chromatin structure. In this part, the genes are all close and packed so closely together, and there's no transcriptional activity. There's no accessibility. So these genes is permanently turned off. A big percentage of the genes are turned off in the cell. For example, in the muscle, all of the neuronal genes are here, they're never been expressed. That's how we control the gene expression. And over here, this is called euchromatin, which is open structure, and this part of the chromatin are open to transcriptional regulation. So when we talk about signal transduction, what do we ever do in the past? Mostly we are studying what's going on here. Now when you have certain regulation, what genes going up, what genes going down. However, if you want to convert stem cell into other cell types during differentiation, or you perform cell reprogramming study to convert cells into one or other cell types, you need to convert these two structures. That means you want to close some of the genes here and open some genes in this part. So this is a challenge which is not well studied in engineering field, even though in biology we know this can be realized by chemical factors and transcriptional factors. But our question is whether physical factors could play a role. So what is the theoretical basis in possibility? Physics or mechanical factor can play a role. For example, we know DNA actually is a polymer of nucleic acid, negatively charged, but they yet organized with a histone as a scaffold here showing that pack around this. And whether this is open or closed is related to its charge and also the modification. And all of these could affect the chromatin organization. So if you look into the cell structure, and here shows this schematic shows how a cell is interacting with neighboring cells or extracellular matrix, and then there will be cytoskeleton connected cell adhesion receptors to nucleus. So for nucleus, we have nuclear laminar. The lamin is the major protein in this nuclear laminar. And there's one interesting finding. The closer the chromatin structure, one unit is unused, they're all packed around the nuclear periphery. Right, so remember, if you go into your walk-in closet and use the clothing, all packed in one place, so you can retrieve them easily. So if you have mechanical perturbation of the cell, there's a possibility that this perturbation, if you deform the nucleus, and this part of the chromatin can be affected, and you have opportunity to engineer the genome. So that was the mechanical view of this epigenetic manipulation. Of course, mechanical cues can also regulate all other cells signaling molecules and regulate the epigenetic state through the traditional chemical signaling pathways. So now I want to show you a couple of examples how we use various mechanical perturbation to enhance cell reprogramming efficiency. And this study was done by Jennifer Soto. She was actually trained at a BNE program here at FIU many years ago. And she did a PhD with me, and now she's a research faculty at UCLA. So in this case, we take a fibroblast from skin, introduce 3 transcriptional factors involving neural development, and then convert cells into neuron. This was developed by Warnick's group at Stanford University. And what we found is the efficiency is quite low in general. If you take the adult skin cells, less than 1% around that range. To improve the efficiency, you can do many different things. When we reprogram the cell, we notice that a lot of cytoskeleton development and cell spreading increase. And then we disrupted the cytoskeleton using a chemical, blackstatin, to disrupt actin-myosin interaction. We found that it's dose dependent. If you increase this chemical, you increase reprogramming efficiency significantly. But if it's too much, it's not good. So there's an optimal level for this disruption. So reducing the intracellular tension in the cell will give some freedom of nucleus and chromatin that can help cell reprogramming. So this experiment shows that the mechanical effect or reducing the tension in the cytoskeleton actually does not induce neuronal genes. If you look into this no treatment control and blackstatin stimulation, the first 2 bars, the neuronal genes showing here, these are all neuronal genes. There's no induction. However, when you introduce the reprogramming factors, BAM, these are 3 transcriptional factors, together with blackstatin, you have significant enhancement. That shows that mechanical tension, in this case, plays a permissive role that facilitates the chemical reprogramming, biological reprogramming of the cell, instead of directly infusing the neuron. So that's one important piece of evidence. So what exactly is going on? And what we found is that when you treat the cells with this blackstatin chemical, not only reduce the tension in the cytoskeleton and allow nucleus some more freedom, you also induce the changes in the nucleus. If you do nuclear staining of this open chromatin marks, this is acetylation of histone 3, the methylation, trimethylation of histone 3, lysine 4 residue, and the monomethylation, H3K4. You see all of these are increased. And also consistently, all the enzymes that result in these changes are changing in the same direction. We don't need to get into detail. So the baseline is that when you reduce intracellular tension, you make the chromatin more open. And we can use the ATAC sequencing to prove that, and directly test the accessibility of the chromatin. In here, showing the 3 examples of neuronal genes. If you look at the peaks, you have more of this opening when you treat the cells with blackstadin and a promoter or enhancer of these neuronal genes. That directly prove physically there's an increase of accessibility. So the other thing we tested is to culture cells on different matrices with different mechanical property. So you don't need to do any cytoskeleton manipulation, and rather grow them on different stiffness. For example, glass, which is megapascal level, or 20 kilopascal, which is intermediate stiffness, or 1 kilopascal, which is soft surface. So surprisingly, what we found is that at the intermediate level of stiffness, we have highest efficiency to convert fibroblasts into neurons. And this is independent of all different kind of matrix proteins. It's not chemical matrix component dependent. It's mechanically dependent. Okay? And it's interesting that this highest efficiency happens at 20 kilopascal. Indeed, if you perform this accessibility assay again, the red color shows more open chromatin. It's consistent with what we found at the intermediate level of stiffness. We have highest accessibility for these neuronal genes at promoter. So that's the effect of matrix stiffness. And again, for chemical signaling, what is correlating is this biphasic induction of reprogramming efficiency is that histone acceleration, which is important for this stiffness effect. And also other specific assay we perform using the chromatin immunoprecipitation and PCR, showing indeed at the promoter of neuronal genes, we have the same trend as these global changes of histone acceleration. Okay, so in the literature, you may find many different kind of reports on the epigenetic regulation by mechanical factors, and some of them are conflicting. And even for what we've shown here, with mechanical disruption or matrix thickness manipulation, we may see different results in different cell types. It's a context dependent, right? How mechanical factors regulate that. And then what we need to do is directly test whether mechanical deformation of nucleus could make a difference in the genome chromatin structure. In the past, there are many systems that have been developed to manipulate cells. It could be single cell or many cells. It could be a micropipette aspiration. You could use atomic force microscope to apply force to the cells of nucleus, or use optical detectors to apply force to the cell of nucleus. In all of these systems, for single cell study, you can also couple this with microscopy and other methods to study a single cell function. And what we decided to do is instead using microfluidic device. In this case, we can have high throughput processing of all the cells, and we can apply different kind of mechanical constriction to cells. For example, it could be cyclic constriction or different degrees of constriction. We apply this mechanical deformation to cell directly in milliseconds, very short time period. And we can see how this will affect the genome organization and the reprogramming efficiency. Now here shows the experimental procedure. We have fibroblasts. We first introduce the transcriptional factors, as we did before, is for neuronal reprogramming. And then we activate these genes in the cell. So all of genes are in the genome. After that, we subject the cells to mechanical deformation. So the hypothesis is that when you deform the cell nucleus, in the presence of all of these reprogramming factors, you will increase accessibility and transport of these factors in the cell nucleus, and that will help improve cell reprogramming efficiency. So that's what we expected. And here shows the one channel. You know, we have cells coming in here and coming out, and we optimize the condition. So we have to customize the channel size based on the cell size. You don't want to squeeze cells too much and kill them. But at the same time, you want to have the channel small enough so that they can apply mechanical force to cells and deform the nucleus. And And we found that if you have a fibroblast with an average nuclear diameter about 10, 11 microns, and if you use 7-micron channels, here it shows, they're not going to kill cells, but you can effectively deform some nucleus. And that's the condition we use. We can use a big channel, 200 micron, to allow cells flow through without any mechanical perturbation to cell nucleus. So after you squeeze the cells, and cells become softer. So you can prove that with atomic force microscopy study. And you see, indeed, the cell becomes softer after squeezing, and then gradually it comes back. The cells will recover with time. And then we examine the neuronal gene expression. With squeezing, you see within hours, you already see the induction of neuronal genes, much more effective than the control, which is not squeezed. And if you look at the neuronal gene promoter-driven reporter, this is also consistent. We have much higher induction of the neuronal genes in the fibroblast. To monitor this temporally, we use a nano probe. In this case, it could detect the mRNA expression of neuronal genes. And here you can see that with time, we monitor the tubulin, the TUB3 neuronal marker. You can see neuronal genes also induce quickly with time, within 12 hours compared to control. And now the question is, what is the mechanism? In the nucleus, we know DNA have condensation. The condensed DNA are close structure, and what we found is we stain for this H3K9 and trimethylation, which is a marker of a closed chromatin structure. You see many condensations in the control group. If you squeeze the cell, this condensation is gone. You become diffuse signal. That means you make this DNA closed structure open transiently, and this comes back after 24 hours. You can also use a FRAG probe to detect this in real time, and this could detect within a few minutes, right? The color of this H3K9 trimethylation decrease. No, it's red at the beginning, and just within a few minutes, when you get out of the microchannel, this becomes green. So this shows indeed mechanical deformation of nucleus induce these chromatin changes, and that is related to chromatin reorganization. And DNA methylation is consistent. If you squeeze the cell, you decrease this condensed DNA structure, similar to the heterochromatin staining. And if you perform the accessibility test, ATAC sequencing, you observe the very interesting effect of mechanical squeezing. So this is genome-wide accessibility. If you look at each row, it represents one gene. Here it represents many, many genes here, and we have cluster analysis. About 1/3 of the genes, after squeezing, actually becomes more closed in the promoter region. And about 2/3 of genes become more open. And here shows a couple examples, neuronal genes. In the higher peak shows more open structure. And this gives us opportunity to manipulate the cells. For example, if we know certain sites are more open, we can design guide RNA using CRISPR gene editing tool to activate those genes more effectively. So we prove this is true. If mechanically you induce this open chromatin structure, we can use that for cell engineering. We compare the biophysical effect with chemicals. And here we have a couple enzymes which can induce the same kind of changes in the heterochromatin modification or DNA methylation. And if you compare that with the physical squeezing, the physical squeezing alone is already effective to induce the reprogramming efficiency. And when you add chemicals, you only have some incremental increase. If you use combination of these two chemicals, they could raise the efficiency similar to physical factor. So in this case, the physical squeezing of nucleus could reach the same effect as the chemicals. And in this, the concept mechanoceutical, the mechanical force could be used as a drug to reprogram cells. So what is the mechanism? When you squeeze the cells, what happens exactly? And we examine nuclear laminar, the nuclear mechanical structure. And what we show is, indeed, you induce some nuclear wrinkling and disrupt partially the lamin structure at the nuclear periphery. So mechanically, there's some effect on the nuclear structure. And if you perform staining, M6 tracer in this case, was stained for the lamin-associated domain of gravity. And lamin here, red color, is the structure laminar. And we found that there's a decoupling of this DNA in nuclear laminar. So here shows the colocalization is decreased if you squeeze the cell. So that proves our hypothesis that if you have mechanical perturbation of the cell nucleus, some of the chromatin structure may be detached from the nuclear laminar and allow more freedom to reorganize. So if that's the case, if you knock down this nuclear laminar structure, will that have the same effect? So we performed study to show, indeed, if you use siRNA to knock down nuclear laminar, right, you can see the decrease of DNA condensation, similar to mechanical squeezing. You also see some wrinkling of nuclear laminar as well. And the efficiency of reprogramming increase if you knock down nuclear laminar here, similar to squeezing. And the combination didn't increase this further, showing that they're exerting the effect on the same mechanism. So finally, we decided to generalize this. We asked the question whether this is true for other cell types. For example, if you take macrophages from circulating blood, which is easy to isolate, and you want to convert macrophages into neuron, do we have the same effect? Right? We squeeze macrophages, we did show that the induction of neuron efficiency is higher if you squeeze the macrophages. We also try to convert the fibroblasts into other cell types, for example, induced pluripotent stem cells. And if you squeeze the cells, the efficiency of converting these into iPSC is also much higher. So that shows this mechanical effect can be generalized to different cell types. We also design different kind of microfluid device to increase the efficiency, increase the throughput to process the cell. So we can go let millions of cells go through a device within minutes, and then we can allow high efficiency of cell type conversion and cell reprogramming using different systems. So that summarizes what we found for this part. It is important for mechanical regulation inside the cell. If you reduce the intracellular tension, you will enhance the chromatin opening and cell reprogramming. And if you manipulate the extracellular matrix, you can also affect the chromatin accessibility in cell culture. And finally, if you directly deform nucleus in suspension in a microfluid device, you can use that to enhance the efficiency of gene editing and cell reprogramming. And with that, I want to thank all the lab members. Jennifer Sato and Yang Song contributed to the mechano-epigenetic engineering product study, and Leo and several other team members contributed to the immune cell engineering project, and all the collaborators and funding sources. Thank you.
Thank you, Dr. Lee, for the presentation. So we have some time to carry out the Q&A. Any questions from the audience?
Start. That's really nice. I'm wondering about the actin-myosin tension and opening up chromatin. I'm thinking about work from like Dennis Disher and Adam Engler that show like maybe different substrate stiffnesses can promote different types of lineages, right? So like osteogenic versus adipocytic. So have you tried to look at that? Can you, by modulating that tension, like titrating at different levels, can you open up different types to different places in chromatin that might favor one cell type versus another?
So that's a good question. The question is whether if you manipulate the tension in the cell, quantitatively titration, and you can induce the same effect of the surface stiffness as what the Dennis Dish and Adam Angler have shown. And indeed, actually, that's something we are testing. This shows that there's one explanation of this cell type specification. This mechanism could be epigenetic, really. And of course, there are many other studies showing the signaling events could play a role. Yeah, so it is possible. But when you just simply disrupt actin-myosin interaction, the effect is still slightly different from the stiffness manipulation. So when you manipulate the cell adhesion, the effect is not all the same compared to the cytoskeleton disruption. Many other things could happen.
Okay.
Yeah, amazing presentation. Very close to science fiction, I could say. So I found answers to many questions that I have in the past, and one of them is during development, embryonic development, how neurons spread, and this is related to a migration process. That migration happens through channels that are formed by astrocytes, glial cells. So I think that those channels probably simulate the microfluid device, and when they are moving from certain areas to the brain where they are going to express, that forces probably differentiate the type of neurons that are ending depending on those channels. So my question is, in this new environment of organs, in which the manipulation is basically chemical, can we start thinking on scaffold that also incorporate microfluids so that we guide those cells mechanically where they are going to be differentiated, not only chemical, in addition to the chemical?
Yeah, that's a good question. Whether we can manipulate the actual set of a matrix in the microenvironment incorporated into, for example, organ-on-a-chip system, or even to understand how this happens during the development. Yeah, actually, this indeed is relevant to some of the in vivo conditions. Development is one good example. And also, during the disease development in cell migration, for example, the cell needs to migrate through a three-dimensional matrix, and then actually the cells are squeezed through. Right? In 3D matrix, the migration is different from 2D. They need to deform. And when the cancer cells, during the metastasis, they need to migrate out of the blood vessel into the tissue, and they need to deform significantly when they migrate through the cell junction in the blood vessel wall. So all of these are good examples. So I think, yeah, it is possible to develop some system to study this further under various conditions, physiological, pathological conditions, and development. And that will probably involve more extensive manipulation, 3D printing, micro-nanofabrication to simulate the environmental factors.
Any other questions?
Very, very great presentation. My question is around the personalized, and you mentioned the cost and all that. So in the T-cell isolation, T-cell manipulation, and then creating the actual antidote for the personalized cancer cell, where is the cost? If one research was going to focus on reducing the cost for accessibility, where is the cost concentrated in that process?
Yeah, definitely, the cell culture condition, all of these should be well defined. All of the components, and also the procedure, the clinical procedure, those will be the major expenses. Yeah, and just the therapeutics itself, if you involve doctors and even some professionals to help for cell manipulation, that will add quite a lot.
I'm sorry, go ahead.
So with the first presentation that you were talking about, were there any relapses in the cancer? Like, did it end up growing back after the 20 days?
Yeah, so that's a good question. So here, the experiment, I just showed the time point up to 4 weeks. If you have a good percentage of T T memory stem cells, CAR T cells. They are going to home to the bone marrow, and hopefully they can have long-term immunity against cancer. They can prevent cancer formation, relapse in this case. So when we perform these studies, indeed, when we have a Simivac for T-cell, CAR T-cell expansion, it has much better effects. I think we should probably perform longer time study to see whether it will prevent its cancer recurrence further or permanently.
I have a question about the CAR T, and I'm not in that area, so my question might not be very intelligent. So I noticed you use bees to activate T cells, and your image, some of the microscopy images show T cells attached to the bees. So in the subsequent steps, how do you separate T cells from the bees, and would T cells stay in the active state, after separation?
And that's a very important question for manufacturing. For our case, the alginate beads can be separated from the cell by a couple of different ways. Because the interaction between T cell and these beads are not very strong. So one is you can use centrifugation to separate the cell and the beads. In a condition it's very mild. And two is, in some case, if you use a viscoelastic beads, because they are cross-linked by calcium, you can use EDTA to dissolve the bead within minutes, and all the beads will be gone, and you can do centrifugation again to remove them.
Any other questions? I have one more about the squeezing, to the cells. So you showed there are different ways to apply mechanical force to the cells, and one is AFM, to use the cantilevers to tap them. But that's more local mechanical force. And so my question to you is, do you need global squeezing or to apply mechanical force globally to see the effect show, or you can just tap a portion of the cell and then to induce a similar effect? And then the follow-up question would be, does the amount of force applied to the cell matter? Does it give you different results? That will depend on the amount of force you apply.
That's a very good point. So one is the point force versus a kind of a global force, right? So AFM itself may not induce the same effect. Firstly, as you said, it's applied at a certain point. Even though you use a kind of a hemisphere shape of the probe, you only affect a part of the cell. And one difference is that you don't have a confinement. Microfluidic device, you confine the cells in all three directions. If you compress in one direction, the cell may extend to other directions, right? It's a Poisson effect, but it's limited. When you have AFM, in the other case, it's an open system. You compress it, the cells may deform. They have other ways to adapt to this applied load. So they may not induce the same effect as on parameter. And especially, there's another factor which is rate dependent. If you apply force slowly, the cell will adapt to this new condition without significant change of parameter. So rate is also important in addition to force. And another point I want to make is that deformation probably is the key. Now, what is the deformation resulting from the force? So that will be probably better measurement of how much effect we have on nucleus. We need to induce the deformation of nucleus. The force, the cells may not sense the force so much, rather they sense the deformation in this case.
Oh, are you sure? Sure. I was gonna ask, so, thank you, by the way. The main, method of applying force to cells was a compressive force, was squeezing it. Is there any, studies into, extending the cell before it, rip? Is that even possible? Can you extend the cell, and do you think that would cause any cause of the nucleus, and therefore the DNA within it, or can you give me your thoughts on that?
Yeah, actually, the study on that, in this case, they use a different approach. For example, you can adjust the salt concentration, use osmotic force to induce swelling of the cell nucleus. And people have tried that. But that actually could induce additional effects. There are some chemical changes, for example, transport and many other things will change. It's not purely mechanical. But there are some studies on that as well.
Perfect, thank you.
Yeah, sure.
Since this study was primarily, like, it looks like for treating cancer, cancers also come in many forms. Is there a more, did it work in a specific cancer of the body, like in a specific organ or cell in the body that you can see that the effect actually works when you're squeezing the cells?
Yeah, that's an important question. So probably it's regarding the CAR T cell, right, in this case. We tested 2 cancer models. One is lymphoma, which is liquid cancer or blood cancer. The other one is ovarian cancer. Ovarian cancer is also kind of a more liquid-like, in between the solid tumor in liquid phase. So in both cases, these CIMVAC expanded CAR T cells work better. But yet we need to test other cancer models, for example, breast cancer, melanoma. In the solid tumor, the conditions might be different. Probably you want to combine that with checkpoint inhibitor, for example, to inhibit the suppressive environment in the tumor and allow these immune cells to be activated and attack tumor cells. Yeah, indeed, that's a very good point. So for different type of tumors, you probably want to combine this CAR T with other therapies.
Thank you.
I have another question. So with regards to the squeezing of each cell, have you considered doing potentially more research into the type of, so you mentioned that there were different types of cells, but does the amount you squeeze each cell, is that the same, or how should I put this? Does squeezing like cell A and cell B to the same degree cause the same effect of DNA change, or like maybe like this cell requires more of a squeeze that induces DNA change compared to a different cell? Have you done any research on that, or is there any research on that?
It's a good question. So that could be extended to several things. For example, different cell types
Yeah.
may have different sensitivity. And even the same cell type, when they're at different phases of cell cycle, the nucleosides and cell size might be different. So this response could be heterogeneous. And that's why in some cases you probably have seen that we induced the efficiency probably 20%, but we didn't induce to 80% or 100%. There are some other cells which may not respond in the way that we expect they respond. Yeah, so there may be some heterogeneity here for the responses.
Perfect, thank you.
Yeah.
Any more?
No? Let's thank the speaker again for the wonderful presentation.